Okay, sir. Now.
Okay, very good evening, my dear friends. Nice to meet you. Okay, this nice evening, our madam is going to explain one of the beautiful structure about the in the part of the embryology. So today our research person, Dr. K.RANI Madam Garu, Assistant Professor, Department of Botany, Government Degree College Autonomous SIDDIPET. Madam, please welcome and start your session.
Thank you, sir. Okay. Good evening, my dear students. Myself, Dr. Rani. So yesterday we discussed that the anther structure. I think in embryology, yesterday we'll start the topic, introduction and structure of the anther. And today we are going to discuss about that the microsporogenesis or male gametophyte or microgametophyte. Okay. So yesterday just to I'll show one or two slides. This is the tapetum slides, Nana. Structure of anther, epidermis, endothecium, middle layers, tapetum. 4 layers are presenting in the anatomy or structure of the anther. So tapetum is of 2 types. In that one is the amoeboid tapetum. This is the types of tapetum. One is the amoeboid tapetum and second one is the glandular tapetum. So you can observe here the arrangement or cells presenting in the tapetal type. Amoeboid means so they are formed from immature tapetal cell. Almost the best example of the amoeboid tapetum is lower dicotyledons and lower monocotyledonous immature plants. And second, glandular tapetum. So these tapetal cells develop from the mature cells of the tapetum. That means all higher plants like many dicotyledonous and higher monocotyledons all belongs to the example of this glandular tapetum. So almost we'll discuss yesterday that function of tapetum. It provides the nourishment to the developing pollen grains. So it helps in the formation of exine and also helps in the transport of food material to inside of the anther. So, and also tapetum helps in the formation of pollen wall. So today the topic is microsporogenesis or male gametophyte or microgametophyte. So generally the microspore mother cell, they will undergo the meiotic division, or before that the development of the male gametophyte. So it is uniform in all the angiosperm plants. Okay, so almost the microgametophyte is nothing remarkably uniform in angiosperms. The microspore, so it is also nothing but pollen grain, Nana. Microspore or pollen grain, both are same. So the microspore, which makes the first cell of the male gametophyte. Okay, the microspore, it is the first cell of the male gametophyte. It will undergo only 2 divisions. Okay, so in that which the result of first division, 2 cells are formed. So in that a large vegetative cell and a small generative cell only. This is the first divisions. So the process of or formation of microspores from a pollen mother cell through meiosis, that is called microsporogenesis. Okay, so the microspore mother cell, so it will undergo the meiotic division and it will forms the from a pollen mother cell through the meiosis, that is called microsporogenesis. So in this, the cells of sporogenous tissue, it will undergo the meiosis to form the microspore tetrad. Microspore tetrad. So they are arranged in a cluster of 4 cells. So I will show the all diagrams one by one. So as each cell of sporogenous tissue has potential to form the tetrad. In this, each cell is microspore mother cell. That means the pollen mother cell. PMC means pollen mother cells or microspore mother cells. They are the completely sporogenous tissue. They has the potential to form the tetrad. Okay. So on maturation and dehydration of anther, the spore dissociate and develop into the pollen grains. So we know that the structure of the anther, this is the This is TS of anther. So the pollen sac in between 4 lobes will, you can see here. So in the almost in the circle of the plants, circle of the part, all the pollen mother cells or pollen tetrads are presenting in the 4 lobes. So they are the pollen grains, they can release with the dehiscence of the anther. So in the dehiscence purpose, which cells are helpful for the dehiscence? Some stomium cells. They are presenting in the almost in the 4 lobes parts, some stomium large cells are presenting in that. So the stomium cells will helpful for the dehiscence of the anther. In that almost the structure of the microspore or other the microspore mother cells, which are at first polygonal and they closely packed gradually become rounded and loosely arranged cells. Just I will show. This is the microsporogenesis diagram. Please, you can see here the microsporogenesis pollen mother cell. So it will undergo the 1 meiotic division. One cell can division to takes 2 cells. After second meiotic division, so the pollen 4 cells are formed. That is the in between, or it will undergo the 2 meiotic divisions takes place, 1 cell becoming the 4 cells. That is the first divisions. And after that the microspore, so that is called microspore tetrad. So that is the tetrad cell. And before that, just we'll discuss that the microspore mother cell undergo the 2 meiotic divisions, gives rise to 4 haploid microspores. So during the meiotic division of microspore, the wall formation may vary in different plants. So in this almost 2 different types of wall formation or cytokinesis have been recognized. One is the successive type and the second one is the simultaneous type. Okay, that means the almost the pollen tetrads or microspore tetrad formation, the wall formation may vary different in plants. So 2 different types of wall formation in that successive and simultaneous. What is the successive type? So in this almost the first nuclear division of meiosis 1. Just this is the meiosis 1 in this type. After the first nuclear division of meiosis 1, 2 daughter cells are formed. So due to the development of the wall formation, that is the successive type. And what is the simultaneous? So after the completion of meiotic division, the microspore mother cell, different type of tetrads are formed. That is the simultaneous. That means 2 meiosis 1 and meiosis 2, 2 divisions takes place at a time. That is the simultaneous. And in the successive, so 1 by 1, just to 1 division takes place after 5 minutes and second division takes place. That is the successive type and simultaneous type. So in the successive type, these nucleus, that means the wall formation, due to the development of wall formation, these 2 nuclei undergo the meiosis 2 and 4 daughter cells are formed in the successive. 1 by 1, meiosis divisions takes place in the successive type, but at a time 2 divisions takes place in the simultaneous type. So ultimately, what is that? Almost the tetrads are formed. The microspore tetrads are formed after completion of the meiotic division. This is the final development or final just to microsporogenesis takes place in the main part. So in this almost so the first division of the microspore gives rise to the vegetative cell or the microspore of the pollen grain. It represents the beginning of the male gametophyte. So the structure of microspore or pollen grain has already they have been given in the almost in this diagram, meiosis 1 and meiosis 2 and tetrad formation. You will observe in this way. So in the almost the tetrads, different tetrads or different types of tetrads are formed in the after completion of the meiotic divisions. They are the tetrahedral, isobilateral, decussate type, T-shaped and linear type. These all are the different microspore tetrads. Tetrads are formed. One is the tetrahedral, Tetrahedral means almost in this type, the 4 spores are arranged in the 4 corners. Just I'll show another diagram. Okay, so in the tetrahedral type, the 4 spores are arranged in the 4 corners. So that is generally presenting in all the dicotyledonous plants tetrahedral type. Okay, and second one is the isobilateral. So in these 4 microspores are arranged in the 4 corners of a square. So same, but the 4 cells are arranged in the square shape. So this is called the isobilateral type. Almost monocotyledonous. Poaceae family are the best example of the isobilateral type is the maize, Zea maize. Okay, and decussate means, so in this type, the 4 microspores are arranged in 2 pairs. So one pair of microspore is at right angle to the other pair. So this is called decussate. Almost all Magnoliaceae family tetrads will observe in the decussate type of the microspore tetrads. And T-shaped, so in this type, the microspores are arranged in pairs. So 2 microspores are arranged side by side, while the other 2 are arranged on one above the other. So it's look like a T-shape. 2 cells are above side by side and 2 are one by one. So one below the one, that is the T-shaped appearance. Almost the best example of the T-shaped type is the Aristolochia. And linear type, so in this type, the 4 microspores are arranged one above the other in a single row, that is the linear. So almost the plants of the family Asclepiadaceae plants we will observe in the linear type of the plants. That is the microspore tetrads. The completely after completion of 2 divisions, we will get that different type of the tetrads are formed. So, and what is the development of the male gametophyte? Okay, so the development of the male gametophyte in angiosperms, almost the microspores represent the beginning of the male gametophytic generation. Okay, so the microspores, they can represent the male gametophytic generation. The microspores, after their release from the tetrads, they are referred to as a pollen grains. Okay, this is the beginning point. The microspores releasing after their release. So from the tetrads means almost we'll discuss tetrahedral, isobilateral, decussate, T-shaped, linear, any one of the tetrad, they are referred as a pollen grains. So the pollen grains are the minute in size. This is the pollen grain, Nana. Pollen grains are minute in size and they can possess haploid nucleus and dense cytoplasm. Okay, so in this, the reserve food materials are presenting in the form of the minute fat bodies. So almost starch or fat bodies are presenting in the pollen grains. So they are the preferred as a reserve food material. Okay, so the microspore nucleus that is pushed towards one side due to the formation of the vacuoles in the cytoplasm. Almost the movement of nucleus that is related to the position of generative cell that is to be formed later. Just this is the pollen grain structure. So after the thickening of the cellulose, exine, intine, nucleoli, some pores, and these all are presenting in the pollen grain. Almost the pollen grain status is, it is haploid nucleus. Okay, pollen grain, they are the minute structures, minute in size, and they will possess haploid nucleus and dense cytoplasm. Some reserve food materials are, they are in the form of fat bodies. Okay, so then after the, what is the structure? The structure of the pollen grain. So it's very, very important, the grain of the pollen. In this, the pollen grain at that time, that is the shed, it is spherical or ovoid cells with 2 membranes. You can observe here 2 membranes. So a rough outer wall of cutin, that is called exine. Just to roughly structure, almost that is completely rough outer layer of the pollen grain structure is exine. Okay, a rough outer wall of cutin. So that is the exine, and an inner thin wall of cellulose and pectin substances. Cellulose and pectin substances, that is called intine. Okay, exine and intine. Outside of the mature pollen, so in insect pollinated plants, there is an oily layer, so which consists of a lipid, carotenoids, and some proteins also presenting in that outer side. Okay, this almost oily layer is called that pollen kit. So here I not mentioned here, just to the outer side of the mature pollen. In the insect pollinated plants, there is an oily layer, so that is called pollen kit. Okay, so some carotenoids are responsible for the yellow or orange color of the pollen, and proteins also make them sticky. As regard its almost the function, it helps in the insect attraction or insect pollination, and it will protect the pollen against the radiations also. That is the complete the structure of the pollen exine and intine. So in some cases, in the pollen grains, we will observe some apertures and sculptures, shape, size, and different chemical nature of the layers also. We will observe that. So in that, the apertures means, according to Edman, there are 4 types of apertures in the pollen grain we will observe commonly. So these apertures are the weak places where exine is absent and the intine comes out as pollen tube. So these places, the pores like structures, almost the exine is not uniforming arrangement of the outer cells. So here and there, some places are weak places are presenting in the exine places. So these apertures are the weak places where the exine is absent and the intine comes out as pollen tube. So in this, the apertures may be simple or compound. Some long apertures also presenting. They are the colpi, C-O-L-P-I. So long apertures also presenting in some different plants. They are the colpi, and short ones are the pores. So hole-like structures, and some pollen grains with simple apertures, either both colpate. So simple and some long, that is the colpate, or some porate with holes also presenting porate. So these are different apertures are presenting on the exine and exine or cell outer wall of the pollen grain. So in some cases, a compound aperture, it consists of a central region, that is the oral, and the outer region, that is the colpal, and colporate pollens also observed in some fossil plants. So these all are some apertures and some sculpturings. So sculpturing means the details of the exine of the pollen, it constitute the sculpturing. So they are the smooth or pitted or grooved or with the projections or some warty, verrucate, or some baculate rod-like elements or some rod-like projections. So some swollen heads, sessile pillar. So some spiny. These all are some different sculpturings also we observed in the pollen walls. Okay. So almost the shape of the pollen, it varies from spherical, subporate, prolate, or perprolate. These all are shapes, and each pollen grain has 2 poles, Nana, at opposite ends of the polar axis. These all are some characteristics and characteristics of the pollen grains. And what is the microspore germination or microsporogenesis? This is the complete spherical in shape, have 2-layer wall, outer hard layer, exine, and inner thin layer is intine. So exine made up of the sporopollenin. It's very, very important in exine entrance point of view. Pollen grain, exine, outer layer made up of a sporopollenin. So it is resistant to organic matter, withstand high temperature, acids, alkalins, and enzymes. So it has prominent apertures, that is called germopores, as I told you. So where sporopollenin is absenting places, that is the apertures also presenting in the exine layer. Intine, so it is very thin, continuous layer. So almost its arrangement is uniformly made of cellulose and pectin. Okay. So pollen grain are male gametophyte or the microspore germination. In simple way, microspore germination. In this, the microspore or pollen grain represent the beginning of the male gametophyte, as I told you. So the structure of microspore or pollen grain has already, it is a newly formed microspore. It possesses very dense cytoplasm with a central nucleus. Just I'll show other diagrams. This is the development of the microspore. And it's a germination, development of a male gametophyte in pollen grains. So the germination of microspore starts while it is still within the microsporangium. Okay, the development takes place within the microsporangium or pollen sac. Okay, microsporangium or microsporogenesis occurring in the microsporangium or pollen sac. Okay, so almost we'll discuss that two cells are formed, vegetative and generative cells. The first division of the microspore gives rise to the vegetative and generative cells. Okay, so the development of a male gametophyte in pollen grain, microsporangium, this is the pollen sac, and microsporophyte, so it is in 2n state. After completion of the two divisions, it will just after completion of the undergo the meiotic division takes place. 4 haploid microspores are formed, so that is the microspore tetrad, and each of 4 microspores, they are in single nucleate, uninucleate, haploid condition. So each of the look like in end stage. After that, the first division of the microspore gives rise to the vegetative and generative cells. Okay, so this haploid cell undergo the division and it forms the 2 cells. 2 means, so gives rise to the one is vegetative cell and another one is the generative cell. Okay, so the first formed walled and they are peripheral cell, that is the generative cell. Okay, so the generative cell, while the larger naked central cell, so it fills the fill of the remainder of the spore wall cavity, that is the vegetative or tube cells. So here I did not mention tube cell. Almost 2 cells, one is the generative and another one is, this is the tube cell. Tube cell or vegetative cell, 2 cells, generative cell and male gametophyte. So this is the nucleus of the tube cell, 2 cells are formed. Okay, the nuclei of generative cell and vegetative cells differ in size. So almost generative cell is small in size and tube cell is large in size. So in that way, they are different in size, structure and in staining qualities also different. So the nucleus of the vegetative cell, it will possesses a prominent nucleus. The cytoplasm of the generative cell is hyaline. That means they are the ragweed. Almost I'll take one example, ragweed pollen grain. So the cytoplasm of the generative cell is the hyaline and it is almost without RNA. Which cell without RNA? The generative cell, almost it is hyaline and almost without RNA. Whereas the vegetative cell is rich in RNA, the DNA contents of both the nuclei are same in the beginning. But later on the increase in the generative nucleus, so almost one have the RNA and both will have the DNA. Content of both the nuclei are same in the beginning, but later only the increase of the generative nucleus. Okay, so there is a considerable variation in the form of generative cell. Usually it is elliptical, lenticular or spindle shaped. This is the generative cell, look like elliptical in shape, lenticular or spindle shaped. So the starch and fat are the most conspicuous substances. They are founding in the vegetative cell. Almost in the formation of generative and vegetative cell, certain proteinaceous bodies have also been reported in some microspores. Eventually, the generative cell, they can loses contact with the microspore wall and it is being shifted into the vegetative cell. So just to events in anther development, longitudinal. So this is the complete, not for that, just you can observe here. So both cells are formed. Continuous missing slide. Okay. So it may be like part of it in the beginning. So in this, almost they becomes oval and spindle shaped. Thus the microspore becomes the two-celled. Generally the microspores, pollen grains are being shed from the microsporangium in two-celled stage. So for pollination, it is only in rare cases, Nana, that the pollen grains microspore becomes the three-celled before its liberation from the pollen sac. It seems somewhat difficult to make clear-cut demarcation between two-celled pollen grains. So because the generative cell, almost some text to information also there. Mature pollen grains has two cells, vegetative cell, generative cell. So vegetative cell is bigger, abundant food reserve, large regular nucleus. Generative cell small, spindle shaped with the dense cytoplasm. And a nucleus is floats in the vegetative cell cytoplasm. So in 60% species, pollen grains are shed into two-celled stage, whereas 40% species shed in three-celled stage, in which generative cell divides mitotically into two male gametes. So this is completion. It seems almost the divisions of the generative cell, where it takes place in the pollen grain or in the pollen tube, occurs in the regular way. Somewhat in this way, almost in the formation of generative and vegetative cells, the generative cell is oval or lens shaped, as I told you, and it is surrounded by the plasma membrane, and cell wall is absent. So the cytoplasm of the generative cell has all cell organelles except the plastids. So all cells are presenting except plastids, Nana. Generally, the pollen grains are dispersed from the anther in these two-celled stage. The pollen grains reach the stigma through the air, water, or insect or animals. So this is called that pollination. When the pollen grains reach the stigma, they observe the secretion of stigmatic surface. So it undergo the swell and get modified in their shape. So during the germination, intine comes out through the aperture or pore in the form of a tube-like structure. This is called the pollen tube. Intine will form the pollen tube. So the vegetative and generative cell enters into the pollen tube by means of mitotic divisions in the generative cell. Two haploid male gametes are formed. So the male gametes enters the embryo sac. The male gametes have a large nucleus surrounded by the cytoplasm. Just another slide also there, I think. Pollination, fertilization. Sorry. Okay, so this is the complete development of the microsporogenesis or microspore development. Two cells, vegetative and generative cells, are formed. So in this two-celled stage, the pollen grains are dispersed from the anther. The pollen grains reach the stigma through the either air, water, insect, or animals. So that is the pollination. When the pollen grains reach the stigma, they can observe the secretion of the stigmatic surface. So it will undergo the swelling and get modified in their shape, different shape. So during this germination, intine comes out through the aperture or pore in the form of a tube-like structure. So that is the pollen tube. The vegetative and generative, both cells entering into the pollen tube. So after that, the mitotic division in the generative cell, two haploid male gametes are formed. So the male gametes enter the embryo sac. The male gametes have a large nucleus. It will surrounded by the cytoplasm. That is the development. And another one. So the structure of the this is the megasporangium or ovule structure. So Just I'll show the slideshow. This is the second part, that is the.
Madam, only 7 minutes is there.
Okay, I will show just diagrams only, types of ovules.
Okay,
Tomorrow we'll discuss clearly the development of megasporangium and examples of the ovule types. So this is the introduction, Nana.
Okay, madam.
Or else any doubt regarding this? Just students are interacted with me, I can stop here. Sporangium, and next one is the megasporangium. Megasporangium means nothing but ovule, the gynoecium or pistil, that is the female reproductive organ of the flower. So in that part, megasporophylls. Okay, so typically the carpel consists of the three parts. The basal swollen portion is called the ovary. So you can just do ovary, and the second one is the median elongated part is called the style. So the apical pollen receiving part, that is known as the stigma. The ovules which are enclosed in the ovary, it will develop into seeds after fertilization. Just this is the parts of flower, almost to sepal, ovule, receptacle, petal, so stamen, anther and filament. This is the up to megasporangium or ovule structure. So ovary is attached to the style, elongated part. So the apex part of the style and ovary both will combine to form the carpel. Okay, so ovules are presenting in the ovary part. Different types of placentation will observing in this in the megasporangium. So this is the ovule structure in separately. The ovule, this is the complete ovule part, Nana. The embryo in this nucellus, 2 polar nucleus, 3 cells, and this is the layers, integuments. So completely polar nuclei, egg nuclei is nothing but female gametes, and this is complete structure is the ovary, style and stigma, you know all these things. So just we'll discuss the types of ovules. So it's very important in entrance point of view. In the many types of ovules are found in the angiosperms based on the position of micropyle. So in relation to the funiculus and other parts of the ovule, 6 types of ovules are recognized in the angiosperm. Comphylotrophus, amphitrophus, sursinotrophus. Okay, 6 types: orthotrophus, anatrophus, hemianatrophus, comphylotrophus, amphitrophus, and sursinotrophus. Just we'll discuss one by one. In this orthotrophus or straight ovule. Orthotrophus, so it is also atrophus, orthotrophus or straight ovule. Completely. So this is the straight ovule of the micropyle. Almost it's look like a straight. So that the funicle.
Madam, your voice is not audible.
So that the micropyle lies close to the hilum. So this is the hilum part. Okay, this type the ovule bends along the funicle so that the micropyle lies close to the hilum. The collagen lies at the other end. This is the commonest type of ovule found both in monocots and dicots. Anatrophus is the commonest ovule. So we are finding in the monocots and dicots. Okay. So in this completely, the best example of the anatrophus, some of the common in Helianthus tridox asteraceae, some of the other common dicotyledonous simply. Okay. And hemitrophus or hemianatrophus. So this is the, in this type, the body of the ovule is straight, but twisted in such a way that is placed transversely at right angle. So this is the funicle, and almost the embryo sac or the ovule body is complete transversely at right angle. And so the collagen micropyle line is at right angle to the funiculus. So it is found in the ranunculaceae and some primula. These all are, the body of the ovule is placed at right angle to the funicle. In simple way, the ovule body or body of the ovule is placed at right angle to the funicle. So they curve up to only 90 degrees. For example, ranunculaceae and primula are some oldest plants almost, they are belongs to this type of the ovule. And Comphylotrophus. So in Comphylotrophus, the ovule during the growth curve, so and it is perpendicular to the funicle. In this type, the transfer ovule is bent round like a horseshoe, so that the micropyle and the collagen do not lie in the same line as in the almost Caparis or Cruciferae or Mirabilis poroclac plant. So these all are example of the Comphylotrophus or curved ovule. In simple way, the ovule during the growth curve, and it is perpendicular to the funicle. The embryo sac is